This is Professor Doherty, and today I will be moving into renal physiology. This is the first of two lectures, and in this lecture we're going to cover kidney function and then kidney structure, which many of you are probably familiar with from anatomy. Then we'll move into the structure of the nephron. Again, many of you have probably seen that before, but we'll do some more detail on the physiology of the nephron. And then we'll take a look at the blood supply that goes to the kidney and comes out of the kidney, and then we'll talk about filtration, reabsorption, and secretion. All right, so with that, let's go ahead and take a look at some of the structure of the urinary system here. A couple things I want to point out, and so you should go ahead and write this down. The parts of the urinary system include the following. So let me get a green pen here. First of all, the main structure is the kidney. Okay, we have two kidneys, one on the right side, one on the left side. The kidneys are located at the back of the abdomen. Essentially, you can see usually just below or next to the rib cage. Because of this, they're right up against the back, and so therefore if you get hit in the back, you can actually hurt or damage your kidneys. Sitting on top of them, just so you know, is the adrenal gland, which you can see here on both sides. That is not going to be part of the urinary system, but I'm just pointing it out. All right, most of the physiology that takes place in the urinary system takes place in the kidneys. So we're going to be focusing on the kidneys throughout this talk. However, I am going to point out the other structures here that you should write down as well that are part of the urinary system. So first of all, kidneys. The second part is ureters, which I'm drawing over here on one side, on the right side. The ureters are simply tubes. So you have, again, one on the right and one on the left. And these are just tubes that transport urine that's been produced in the kidneys down to the bladder. And so that brings us to our next part, which is the urinary bladder, which is right here. And that is simply a storage point for urine. All right. Now notice also in this picture that this is a female, and right behind the bladder is the uterus. And so when the uterus begins to expand during pregnancy, guess what it pushes on? It pushes on the bladder. And this is one of the reasons why pregnant women have to go to the bathroom so often, is because this pressure coming from behind, from the growing fetus, is basically going to try to shove that bladder shut. Okay, and then coming out of the urinary bladder is just another tube here, and again, this is the female version, right? The urethra. Males also have a urethra, and basically that shunts blood or urine out of the body and out of the urinary bladder. The other parts that I would like to point out here that are part of the urinary system as well are the blood vessels that go into and out of the kidneys. So if I'm going to clear these drawings. So you can see here, this is the renal artery here, and there's another renal artery heading back behind the inferior vena cava there. The renal artery, so here's the name so you can see where it's pointing to. The renal artery brings oxygenated blood into the kidneys, and it's also the blood that's going to be filtered by the kidneys. Now, what often happens here, that's kind of unusual, is that the renal artery actually splits. I know it's difficult to see in this picture, but it splits into a number of different branches before it even enters the kidney. And I've seen this over and over again when I've looked in cadavers, so don't be surprised if you actually see that. It's quite normal. They're all just part of the renal artery. Okay. To go along with that, blood that comes out of the kidneys comes out through the renal veins, and you can see the renal veins also have a tendency to branch as they come off, and then this returns blood into the inferior vena cava that's then going to go back and get processed by the heart or pumped by the heart up to the lungs to be reoxygenated. All right, so those are the parts of the urinary system. So you should have kidneys, ureters, bladder, urethra, and the renal artery and renal vein. Now let's talk real quickly about the functions of the kidney. Now, the functions of the kidney, you should understand that they all basically, all the functions are really around maintaining the blood. Okay, so the blood and urine through the kidney are connected. So really the kidneys are there to filter blood, all right? Now, one of the things they do is the kidneys regulate urine production. And they do this by looking or by changing the water volume of the blood. So this affects the water of the blood. That's what this is talking about. Okay, so for instance, if the kidneys decide that your blood volume is too high, your kidneys will pull more water off the blood, and therefore you're going to have more urine. As this says here in this picture, let me change my ink color real quick. The water that's pulled off of the blood is going to affect the plasma volume. So as you pull more water out of the blood, your blood plasma volume is going to decrease. And if you leave more water in the blood, that's going to increase plasma volume. Okay. Plasma volume in turn is going to affect blood pressure. So all of these three things are tied together essentially. So the point I'm trying to make is that the kidneys can actually have an effect not only on the volume of your blood, but also on the pressure of your blood. And they do all of that through controlling water. So as kidneys pull more water out, that's going to decrease blood volume, decrease blood pressure. As they leave more water in the blood, that will increase blood volume and increase blood pressure. Okay, next thing, and that's all part of regulation of urine production, all right? Next major thing that the kidneys do is they excrete, and specifically what they're designed to excrete is nitrogen. So we're really looking at getting rid of nitrogen. Our nitrogenous waste comes in the form of urea, and urea has this molecular formula over here, and you can see that it has two nitrogens in it. NH2 to the second means that there are two nitrogens in this molecule. If you look at this by weight, then nitrogen is 46% of the overall weight of this molecule, so about half the weight. So that's a good molecule for us to get rid of. Now, the reason that we have to handle nitrogen in this manner is because nitrogen is non-volatile. So think about our waste products when we do metabolism. The big one is carbon dioxide. Carbon dioxide is volatile, meaning that it can become a gas, which is great because then we can just breathe it out. So carbon dioxide is very easy for us to get rid of. Nitrogen is also a waste product. It's usually a waste product of burning fats, but because it's non-volatile, we can't get rid of it in a gaseous form. So because of that, we have to put it in a liquid form, and our liquid form is basically urea that is dissolved in liquid. So here we actually have to make a fluid using water in order to get rid of urea. Now let me say this, that's actually kind of bad because we actually use a decent amount of water just to get rid of this nitrogenous waste, this urea. It would be much better if we didn't have to use so much water to do that. If you think about humans in an arid environment, the last thing you want to have to do is pee in order to get rid of urea when you really need that water if there's no water around you in a desert environment, right? Now, that's how we get rid of it. So we get rid of urea in the form of urine. So we dissolve it in water, but other animals don't do that. Not all, at least not all animals do. If you look at, for instance, birds, you ever notice that birds have poop that looks white like this? And yeah, I chose this one because it makes a little smiley face. So this is bird poop. And if you've ever looked at bird poop, you've seen, we've all had it on our windshields, right? You've seen that it's white. The reason that bird poop is white is because birds actually combine their poop with their nitrogenous excretions. And you might have, I don't know if you ever noticed this also, but birds don't pee. And the reason they don't pee is because they put their nitrogenous waste in with their poop. That turns their poop white, their nitrogenous waste does. I don't recall if they use urea or not. I think they might use a different molecule, but it's very similar. But basically, this is how they're getting rid of their nitrogenous waste, is by pooping rather than peeing. There are some animals also that, like desert animals, like I think there are some desert shrews that in fact don't pee at all. And instead what they do is they just put out pellets of a molecule that's similar to urea. So they don't even combine it with water. The reason we combine ours with water is because urea can be toxic unless it's combined with water. So they just have what I consider to be a better system, basically. Okay, next thing that kidneys do is they maintain electrolyte balance. And of course, all of these functions are important. This one is very important as well. And when I say the electrolyte balance, I'm talking about the electrolyte balance of the blood, right? So all of these functions are related to what's happening with the blood. So in other words, the kidneys can take away any of these molecules that we might have too much of in the blood. So if you have too much sodium in your blood, guess what? Kidneys are going to pull that out and put it in the urine. Got too much potassium in your blood? Kidneys are going to pull that out and put it in the urine. Got too much bicarbonate? Same thing. Got too much hydrogen, so your pH is too low. Kidneys are going to pull it out and put it in the urine. In other words, the urine is basically a dumping ground to maintain balance and homeostasis of the blood. Okay, so our body cares about homeostasis of the blood. It does not care about homeostasis of the urine, at least for the most part. There are some regulations on that, but usually we can dump whatever we want into our urine to maintain balance of our blood. Okay. All right, next thing it does is it regulates pH and acid-base balance, and that relates to these electrolytes that we were just talking about. So the two important ones here are bicarbonate and hydrogen ions. So of course, hydrogen is directly related to pH. So the more hydrogen ions that you have in your blood, the lower your pH is, and vice versa. And of course, this is extremely important. We've talked about how it affects proteins and a whole bunch of other structures as well. And then finally, the kidneys help to regulate the blood by producing erythropoietin. And so we've talked about this. This has a name of EPO, right? That's its shorthand name. There's an artificial version of this made by a company called Amgen up in the Bay Area. And erythropoietin, of course, causes red blood cell production in the bone marrow to increase. Now, let me talk about artificial EPO for just a moment here. So, artificial EPO made by Amgen was originally designed as a way to increase blood volume and blood cell count in people who had anemia. So, it was used therapeutically. But of course, we all know that having more red blood cells means that you have more hemoglobin. Having more hemoglobin means that you're able to transport oxygen better. And so athletes got a hold of this and began injecting themselves with EPO in order to increase their athletic performance. Probably the most famous person that did this is Lance Armstrong. He was a guy who, if you're not familiar with him, he was a guy who won like 7 Tour de France, the bike races in France, back in the early 2000s. He was accused of cheating using different drugs, one of them being EPO, but they could never catch him. And so he won all these Tour de France's, and the reason they couldn't catch him is because in blood there was no test for natural erythropoietin and artificial version of EPO. So he won all these Tour de France's, he retired. Years later, someone developed a test for artificial EPO in blood. So what the Racing Federation did is they went back and they tested all of Lance Armstrong's old blood samples that they still had sitting in a freezer someplace. And guess what? They were all hyper high on artificial EPO. So basically they could prove that he was cheating. They went back and stripped him of all of his titles. So that's just the most prominent example of how these drugs can be used to cheat. Now I want you to think for a moment, what would be the danger of using artificial EPO? You can pause this here if you want, because I'm going to tell you the answer. The main danger of using artificial EPO is that you can die from it due to an increase in the viscosity of your blood. So if you think about it, if you use artificial EPO, you're artificially bumping up your red blood cell count, which means that you are artificially increasing your hematocrit, right? So remember, normal hematocrits, let's say for a male, are somewhere around, let's say 50%, right? So that's a normal hematocrit amount. If you use EPO, well, you can maybe bump this up to maybe 55%, right? Or maybe even higher than that. The problem with that is that the more of your blood that becomes red blood cells, the more viscous it becomes, the thicker it becomes, in other words. And at some point, there's going to be a point where your heart can no longer push that thick, viscous blood around, and it just stops. So it's actually quite dangerous to use artificial EPO. The way that Armstrong got away with it is he had a physician who was directing him. So there was an Italian physician who was in on this thing. It's called blood doping. This is one of the ways of blood doping. So this physician was helping him with the amounts that he should take and when he should take them, and not in order to not get caught. And so, not to say that he wasn't, a great athlete, but he was just cheating on top of it. So, all right, with that, with my little Lance Armstrong rant, let's go ahead and take a look at kidney structure. And so now, let's go ahead and do a drawing here. So this is going to be a drawing of kind of the macro portion of kidney structure. Okay, so you can go ahead and follow along and do this drawing. We're going to draw the outline of a kidney here. And the first couple things of the structure of the kidney that I want to bring in here are the arteries and veins. So we talked about these before, so this is going to be, give this a little bit of color here, you don't have to. This is going to be the renal artery. And then, so that's going to be bringing blood in, right? So blood flow coming in here. And then we've got the renal vein. That's going to be taking blood out. So blood flow is going to go out here. And then we actually have a third structure here that I'm going to do in green and actually get rid of a little bit of this. And this is going to be our ureter. So this is going to take our final product of urine. Urine's going to come in here, and then urine will travel down the ureter, and eventually it's going to go to, down there somewhere is going to be the bladder. Now, one of the downsides to the ureter is that it's a rather thin tube, and if anything gets stuck in there, it's incredibly painful, apparently. So you may have heard of like kidney stones. Kidney stones are when somebody gets a buildup of basically a type of calcium in the kidney. It turns into kind of like a crystal-like material, and then if that breaks free and gets into the ureter, apparently it's like the inside of the ureter is like really sensitive. And so it can be very, very painful if somebody has that happen. So best way to avoid that, drink plenty of water. Okay, so within the kidneys, there are structures here that I want to draw. So go ahead and just follow along with this drawing. I'm going to draw maybe 5 or 6 of these. We'll see how many I can fit here. I'm going to draw them a little taller on this one. Okay, so these things, which I kind of drew as blocks, but they should look more like pyramids. These are called renal pyramids. They're called this just due to their shape. They typically have this kind of triangular shape, more like the one down here. These renal pyramids basically have a bunch of tubules from the nephron in them. Not that you need to know that at this point. But these renal pyramids kind of sit in the center area of this kidney. So let's describe these areas. I'm going to highlight a couple areas and then remove that highlighting. The center of this organ, which I'm going to color here, so this entire yellow area here, basically from the top of the renal pyramids down into the center here, is called the renal medulla. Okay, I'm going to remove that coloring and then use this pen here. So basically from the top of the renal pyramids all the way down to the center, just like I colored before, so that entire yellow area that I covered before is called the renal medulla. Oops. The renal medulla, if you may recognize the term medulla, medulla just means the center of an organ. It's a general term. Renal means kidney, so the center of the kidney. It basically just encompasses this area that has the pyramids as well as this center kind of area here. Now there is another general area of the kidney, and that is this outer portion here. So I'm going to color this part out here so you can see it, and you can color it on yours, and then I'm going to take that color away so I can label it. This outer portion shown here is the renal cortex. All right, so I'm only going to label part of it here, so I'll do this in black. So from here all the way down to the top of the pyramids, this is the renal cortex. Okay, and cortex you might recognize as another general term. It basically just means the outer portion of an organ. All right, so with that, let's go ahead and now talk a little bit about the structure of the nephron. So I want you to see a nephron here, just kind of in general. I'm going to draw it large, so larger than it should be. So a nephron is going to sit right about here, and a nephron is the structure that actually does filtration. So notice I started off with a half circle, I did some squiggles, then I'm going to drop this down and bring it back up, and then do some more squiggles, and then have an ending portion over here. Now what happens in this nephron is through the series of tubules, so these are actually a series of tubules which we'll draw in just a moment here. It's going to do what's called filtration, and then secretion and reabsorption. So it's going to do all three of those things, and I'll describe those later. But the way that it does this is this blood supply coming in from the renal artery is going to continue up through this area here, and then it goes into the nephron here and goes through a small little capillary bed and then comes back out. Okay, and filtration is actually going to take place in this area of this capsule and this small capillary bed. Now, one other thing I will mention here is that I mentioned that this vessel runs through this space here. This space between the renal pyramids here and all the spaces, so I'm just showing you one here, is called a renal column. And basically the renal column is all of this space between the renal pyramids. So this is a renal column. All right. And of course every space between these, so this would also be a renal column over here, as is this one, as is this one, right? But this is where the blood vessels actually pass between the renal pyramids, which is why I drew the blood vessel in here. Again, so the renal artery brings in this blood. It goes through several different types of vessels, or different names of vessels, and then eventually the vessels wind up in here to be filtered through a nephron. So let's go ahead and label this. I'm going to do my yellow color again. So this entire structure here, this black structure that I've drawn, this is a nephron. And this is the functional unit really within the kidney that does filtration. All right, so now let's go ahead and take a look at the structure of a nephron. Oops, go back to my black pen. So nephrons, like I said, are these small filtration units that are found in the kidneys, and there's thousands and thousands of these things. But this is where the actual physiology of the kidney takes place. Okay, so let's go ahead and I'm going to blow up that drawing that we did earlier. So here I'm going to make that half circle that I was talking about before. Coming off of here is a tubule, and I'm not going to draw it as a full tubule. I'm just going to make some loops here and then bring it down and up, and I'm going to do some more loops over here and then out to the final ductwork over here. Okay, so let's go ahead and label the parts of this. So we're basically going to do the structure here. But first, actually, let's go ahead and bring in the blood supply here. So here's blood supply coming in here. We go through, this is basically a capillary bed in here. And then the blood supply comes back out. Okay. So this blood supply, notice that I drew it all in red. It's because it's all oxygenated. And so the blood supply coming in from here, this arterial, because this is an arterial, is called the afferent arterial. This arterial is going to bring blood supply into our capillary bed up here. Now, after the blood has been filtered, it then leaves through another arterial called the efferent arterial. So the exiting arterial is called efferent. Notice that both of these are arterial, so that's kind of unusual for a capillary bed. Normally you would expect a venule on the exiting side. Here, so the reason I'm pointing that out is because here in this capillary bed, there's no gas exchange. So we're not exchanging gas here. We're just doing filtration of the blood. All right, now this whole unit here has several names, okay? So first of all, the entire thing here, so both the black outer coating as well as the inner part, is called the renal corpuscle. So the renal corpuscle. The renal corpuscle has two parts. The outer part, which is shown here in black, so let me highlight that. And so you can label this independently if you want to. This outer part is called the glomerular capsule. Now, the other part of this, so capsule, did I spell that wrong? I think I accidentally added a T there. Then the inner part of the renal corpuscle, so this is the second part of the renal corpuscle, is called the glomerulus, and that is this part that I just highlighted in yellow here. So it's basically that capillary bed. So the renal corpuscle is the glomerular capsule plus the glomerulus. And the glomerulus is the capillary bed. The glomerular capsule is that capsule in black on the outside. Now there's a couple different names for the glomerular capsule. You may have heard it called the Bowman's capsule. That's fine to use that terminology as well. All right, so those are the structures of this portion of the nephron. The next part of the nephron is this part with all these kind of squiggly tubules. So let me describe what happens here is urine is going to be filtered, or pre-urine I should say, is going to be filtered off of the nephron and move into these tubules over here. These tubules have a set of names. The name of this first squiggly set of tubules is called the proximal convoluted tubule. Also known as the PCT. Okay, so proximal convoluted tubule. The pre-urine is going to travel through the proximal convoluted tubule and then drop down into this basin down here. Basically, this is all just tubules, right? This set of tubules down here is called the loop of Henle. So both the downside and the upside is called the loop of Henle. And there are two parts to the loop of Henle. There's the descending side, which is going to be here, and then the ascending side, which is over here. Descending just means going down, ascending means going up. So in this case, as the pre-urine flows through the tubules here, it would travel in this direction, and then that pre-urine is going to come around here and then travel back up this other side. Okay, so those arrows are just showing the direction of travel of the pre-urine through the tubules here. All right, then over here we have another set of tubules that the urine travels through. This area is called the distal convoluted tubule. Also known as the DCT for shorthand. And then finally, the urine reaches this final tubule over here. So it comes through this tubule, and then it's going to go down here. This tubule here is known as the collecting duct. Now, this is the one part of the nephron that is not just part of this individual nephron. So the collecting duct will actually collect this pre-urine from a whole bunch of distal convoluted tubules of other nephrons. So if I were to draw this correctly, you would actually have another distal convoluted tubule coming in from over here from another nephron, and maybe another one over here from another nephron that's sitting over to the side over here. Typically, we don't draw that, but I just want you to understand that the collecting duct is basically shared between many nephrons. Okay, we still consider it part of the nephron structure, but it's a shared structure. All right, so with that, I now want to define a few words that are important for our language as we talk about the kidneys. So here's some terminology that's important for you to know. The first term that you should know is filtration. Filtration only takes place, so only happens, I'll put, in the renal corpuscle. Okay, so filtration only happens in the renal corpuscle. So filtration is taking place from the blood moving through the glomerulus. So in other words, like if I redraw like in a small form over here, my nephron, right? So here's my nephron, here's my blood supply coming in. Filtration is only happening here. That's the only place in the nephron that filtration happens. And so you should be very clear about that. And what's happening is that blood is coming into the nephron here and moving through the glomerulus and being filtered across this gap in here, this tiny gap that you might be able to see there. So the blood gets filtered through the glomerulus, and then that comes out as a pre-urine that then starts traveling through the tubules here. Okay, but that filtration is taking place in the renal corpuscle. Second term that's also very important is called reabsorption. Reabsorption is when we take something that's in the filtrate and put it back into the blood. Okay, so taking, I'm just going to call it something because there's lots of things that it does this to, something from the filtrate, and you should understand that filtrate means pre-urine because it's not urine yet. So we're taking something from the filtrate, which is the pre-urine, and putting it back in the blood. So, in other words, let me describe this here. And this is something that I haven't told you about yet, but there's basically a bunch of blood vessels that sit near or on top of the loop of Henle as well as the distal and convoluted tubule. So imagine here we have blood supply that is flowing. It's near the loop here. I'm going to use the loop as an example. Let's say within our filtrate or our pre-urine we have too much sodium. Okay, so we have too much sodium. We don't want to get rid of all that sodium in our filtrate. What we can do is we can reabsorb that sodium from the filtrate through the walls of the tubules here, so it goes back into the blood and gets taken away. That is called, so this process here where we're taking something from the urine or pre-urine and moving it back into the blood, that is called reabsorption. All right, so hopefully that's clear. Reabsorption only occurs in the tubules. So reabsorption will occur in these areas. Okay, notice reabsorption does not occur in the renal corpuscle, but it can occur in the proximal convoluted tubule, distal convoluted tubule, loop of Henle, and collecting duct. Okay, so reabsorption is very common, is what I'm trying to show you here. It occurs in a lot of different places. So that is pulling something from the filtrate back into the blood. Common things that we do this with, for instance, are sugar. So sugar will actually come off in part of our filtrate. We don't want it going out in the urine, so we recapture it. Okay, and that recapturing process is called reabsorption. Final word here is secretion. And secretion is the process of putting something into the filtrate from the blood. Now, this only occurs, so this is not filtration. It might sound very similar to filtration, but this does not occur in the renal corpuscle. So, but not, so we're going to put not renal corpuscle though. This can occur though, where this does occur is it occurs in all the tubules. So basically all of these areas here that were the same for reabsorption can also do secretion. So secretion though is, so if I were to draw this, is, let's say we're over here now. Let's say over here I have, again, we'll use potassium, let's say. Let's say I want to take some potassium and I want to get rid of it. And it appears in my blood supply. So this is in our blood supply up here. So again, we have blood. If I want to get rid of some more potassium from my blood that didn't come out in the process of filtration over here, I can then do what's called reabsorption and take that and put it into the filtrate, and it will get taken away as part of the filtrate and go out in eventually what we call the urine. Okay. This is the process of secretion. So basically, after filtration, we can do two things. We can basically pull stuff out of the filtrate, out of that pre-urine, or we can put more stuff into that filtrate. In other words, this process is more complicated than just filtration. There's a number of other processes that happen here also. Okay, so now I'd like to talk about renal blood supply. So on your paper, you might want to leave yourself a little bit of space where you can do some writing as well as some space for a drawing. So I'm going to do my drawing over here to my right. And I'm going to add some renal pyramids here. Okay, so here we have our blood flow coming in. This is the renal artery. And then we have our blood flow going out, which is our renal vein. Okay, so let's talk about some of the vessels here. So again, we're going to entitle this renal blood supply. So first, let's talk about blood flow coming in. Blood flows into the kidney, starting with, number one, the renal artery. The renal artery is coming off of the abdominal aorta. And what I'm going to do here is I'm going to label this with a green number 1 here. Okay, so I'll turn this green as well. So green 1 is renal artery coming into the kidney, supplying it with blood. Next, the blood moves into a set of arteries called segmental arteries. I'm just going to put an A there. Segmental arteries are in this medullary part of the kidney, so I'm going to label a number 2 here. So essentially, this area here is where the segmental arteries would be. From there, and remember, this is continuous blood flow. There's nothing really blocking these arteries between each other. From there, the third set of arteries that this flows into, or the blood flows into, are called interlobar. The interlobar arteries are found within the renal columns. Oops. And so if we label that over here, it would be a number 3 that's within the columns here. So if you recall, the renal columns are these spaces between the pyramids. So these interlobar arteries are found in these spaces. And again, this is still oxygenated blood. Fourth step is the blood flows into what are called arcuate. So arcuate arteries, like I say, the A that I'm putting here just means arteries. These arteries are, I'm going to put a label number 4 here. Just to be clear, these are the arteries that flow across the top of the renal pyramid. So if I did an example of this, the arcuate arteries over here in green are flowing in this direction like this. They're not the little nubs coming up, okay? So it's the flat red line here. It basically goes right across the top of the renal pyramid. The way that I remember the name of these is arc. So the arcuate arteries arc across the top of the renal pyramids. And then the blood flows into another smaller set of arteries. And here's where it gets a little confusing. These are called intervertebral interlobular. So interlobular arteries. Now these are these tiny little spikes up here at the top of the arcuate. Okay, those are interlobular arteries. Now from here, the blood flow actually then goes into the nephrons. So in fact, there's kind of a jump here. So I'm not going to do nephron blood flow right now, but what you should do is draw something like this, an arrow. So there's a bunch of steps in between here that I'm skipping that we'll go over in just a little bit. But from the interlobular artery, the blood flows into nephrons. So this is where nephron filtration takes place. And there's a set of very small arterioles that we'll go over, but I'm going to skip over that for right now and come back to it in just a moment. Instead, what happens, well, after the blood flows through the nephron, so understand that from the interlobular arteries, so these little guys, we're then going to go, if you remember the glomerulus, we're going to wind up in the glomerulus getting filtration, and then the blood's going to flow out of there and around the nephron. And then from there, the blood's going to come back out. So now the blood has been deoxygenated. And so here, I'm going to use the green numbering again. But we basically just reverse the steps back out. And so the blood coming out has now lost its oxygen after flowing over the nephron. And so the blood coming out is deoxygenated. I'm going to use blue pen here. And the first set of veins that it flows through is called the interlobular veins. So if I were to put the number here on the veins, those are going to be these little guys. And actually, maybe I should use a different numbering set so people don't get confused. Let me call this number 6 instead. So I'm going to call this number 6. These little guys poking up here are the interlobular veins. Then the blood flows into the arcuate vein, so we're basically just going to follow the same path out here that we took in. So arcuate veins, which is then going to be number 7. So number 7 means that it's going to be the arcing. So again, these are arcing over the top of the renal pyramids. Then we're going to move into interlobar. Oops. So interlobar veins, and so those are going to be here. Again, these are going to be within the renal columns. Then we flow into segmental veins. Whoops, again. And so segmental veins are going to be down here also, so number 9 here in blue. And then lastly, we're going to exit the kidney out the renal vein. And of course that's here. And then from there, this deoxygenated blood is going to flow back into, this goes to the inferior vena cava. All right, so that's blood flow in the kidney, so you should understand this blood flow into and out of. Now next, what I'm going to do is I'm going to go through the blood flow in this space in between here. So in this space between the interlobular arteries and interlobular veins, the blood flows around into and around the nephron, and so that has its own set of steps as well. So I'm going to do these mini steps now. So let's clear. All right, so if we draw a nephron here, so here what we're going to look at is nephron blood flow. So after the blood comes into the kidney, it's then going to go through the flow that we looked at for the arteries, and then it's going to do this in red. The blood's going to exit the interlobular arteries. If you recall, that was like our step 5 on the last picture. So the blood is exiting the interlobular arteries here and heading up towards our artery here. What it does then is it enters this set, and we've talked about this artery before. It enters the afferent arteriole. Okay, so let's go ahead and write that over here. So number 1 is going to be that the blood flows into the afferent arteriole. So notice that these are now arterioles, whereas before we had arteries. So these are smaller essentially. So this is going to be number 1 over here. All right, so the blood is flowing in this direction, coming into and towards our nephron. From there, the blood is going to flow into the glomerulus. And our glomerulus, which I'll number here in green, is going to be this squiggly set of lines in there. Okay, and this is a capillary bed, just to remind you. So this is a capillary bed. And remember that filtration takes place here. So we're getting filtration of the blood across the gap here. So the red area is the glomerulus, and basically we're having filtrate coming off over on this side where these little blue arrows are that I'm drawing. And there eventually that filtrate is going to flow into the tubules over here that are part of the nephron. But here we're focusing on blood flow, so let's stick with the blood flow here. Third step is the blood is now going to come out of the glomerulus, and it's going to flow into the efferent arteriole. Now, the efferent arteriole is over here on this side where I'm going to put a number 3. And notice that it's still an arteriole, and it's still an arteriole because the blood was not deoxygenated in this capillary bed. So that's an unusual thing about the glomerulus. It's a capillary bed that does not pull out oxygen. It pulls out instead waste products, this filtrate. Our blood flow in this case coming off the arteriole is still heading in this direction. So it's heading now away from the glomerulus. Next, we're going to flow into another set of capillary beds. So number 4 here is going to be the peritubular capillaries. Now, all the capillaries that I'm going to talk about are in fact peritubular, but I'm going to give them some distinction. There are some different names for parts of these peritubular capillaries. This particular peritubular capillary set is around the PCT, so this surrounds the proximal convoluted tubule, so the PCT. So the way that I'm going to draw this over here on our picture is I'm going to draw it as basically just a set of lines like this to represent that there is a capillary bed here. Okay, so this would be number 4. So we have a capillary bed over and around. So I'll bring this down into this capillary bed basically. So over and around our proximal convoluted tubule is this capillary bed called the peritubular capillary. Next, from there, this blood continues to flow, and it flows into a capillary bed called the vasa recta. Now, the vasa recta is really a continuation of the peritubular capillaries, and you may hear it called that. But I like to split it out because it does have a slightly different function. And the vasa recta actually surrounds the loop of Henle. So vasa recta surrounds the loop of Henle. And here we are starting to get a transition from oxygenated to deoxygenated, but I'm going to keep it as oxygenated. So basically, the vasa recta is going to overlay the loop of Henle. Okay, so these are basically going to be flowing down into here. Of course, it doesn't look like this. This is just a not very good representation, but it's just to give you an idea of what's happening here, right? So number 5, the vasa recta are the capillaries that overlay the loop of Henle. From there, the blood then flows into another set of capillaries, once again called peritubular. But these are the peritubular capillaries that surround the distal convoluted tubule. So the DCT, so distal convoluted tubule. If we look at that over here on our picture, I'm going to do that as a blue overlay because by now the blood is starting to become deoxygenated. And basically these continue up into the capillary bed over here. And so this capillary bed is going to be number 6. I'm going to label it as, so I'm going to give it a blue number here, that's fine. But it's basically this number over here for peritubular capillaries. Then the blood finally flows out into, and so let's give it its exit here. It will exit into the interlobular veins. And so, basically that would be a number 7 someplace over here off the side of our picture, but I'm not going to draw it because technically the interlobular veins are not on the nephron. So there you go, there's the blood flow around the nephron. The reason I'm showing this is because it actually is very important for the functioning of the nephron. So just to step through this real quickly again, the blood is going to enter towards the nephron here, coming from the interlobular arteries. It's going to flow through then what are called afferent arterioles, which is a bunch of split out smaller arteries going to the glomerulus. Filtration is going to take place here within the glomerulus. And then the blood flows out through the efferent arteriole and then into the peritubular capillary bed around the proximal convoluted tubule, then into the vasa recta, which is a capillary bed over the loop of Henle, and then it flows into another peritubular capillary bed over the distal convoluted tubule before it flows out to the interlobular veins, and then of course eventually it's going to flow out of the body. Now there's a couple things about these, and unfortunately I don't have room here, so I'm going to have to erase this and do another sheet here real quick. But there's a couple of different things that these capillary beds do. We talked about what the glomerulus does. It does filtration, but the peritubular capillaries and the vasa recta also do something slightly different. So let's take a look at that. So let's talk about what the peritubular capillaries do. The peritubular capillaries are primarily for reabsorbance. In other words, most of what happens in those areas is pulling, molecules, so substances from the filtrate back into the blood. If you remember, that was our definition for reabsorbance. So the fact that these peritubular capillaries are sitting right next to the tubules makes it so that they can basically suck stuff out of the filtrate, which is our pre-urine. So notice I use the term filtrate for pre-urine. They can pull things out of that filtrate and back into the blood. They can also do secretion as well, but the main thing they do is reabsorption. Now the vasa recta, although it is also heavily involved in reabsorption, one of the main things that's used for is maintaining a concentration gradient. And this concentration gradient is used to pull water out of the filtrate. So really the vasa recta is mostly used for water reabsorption. And it does this by utilizing a concentration gradient. And so that's why we focus on the concentration gradient here. Now I'm going to come back around to these peritubular capillaries and vasa recta when we talk about the physiology of the nephron a little more deeply. But for now, I'm going to move into what we call micturition. So the term micturition simply is a fancy term for urination. But you should be aware of this since it is the official term. Really just means urination. And the control over urination actually takes place because of a spinal cord reflex loop, actually. And there's a couple parts to this that you should be aware of. So part of this is under involuntary control, and part of it's under voluntary control. So in other words, when you urinate, there's a portion of it that you can't control and a portion that you can. Of course, you're aware of the portion that you can because you might get the urge to urinate, but you don't immediately evacuate your bladder. Okay, so there are really two parts to micturition, like I was saying. The first one is the involuntary control. Which is going to be number one here. And that is controlled by a spinal cord reflex. This spinal cord reflex controls what's called the urethral sphincter. And it's actually the internal urethral sphincter, so let's make that distinction. So the internal urethral sphincter, this is a muscle that's just under the bladder. So it basically sits around the urethra. I'll draw a picture of this in a moment. Actually, I might be able to fit one in over here. Let me try. So if we have a bladder here, so let's do this as a bladder. And let's get rid of some of this other stuff up here. Okay. Actually, if I'm going to do that, might as well go all the way here. Let's just do the picture up here. Okay, so we have a bladder. And then a urethra coming off that bladder. Okay, so this is bladder. And here we have the tube here is the urethra, which of course leads to the outside of the body. There are actually two muscles that control the release of urine. The internal urethral sphincter sits about here. Okay, so these red lines represent the internal internal urethral sphincter. And then there's actually a second muscle that is under voluntary control that's down here. And this is the external urethral sphincter. So here the spinal cord reflex controls the internal urethral sphincter. And what it does is it controls this through a feedback loop. So let's say here there's a feedback loop that goes from the bladder and the base of the bladder. So essentially down here, if you remember, there's a region down here at the base of the bladder called the trigon. And basically in this region here, there are stretch receptors. So what happens here is, and I'm going to list this under number 1 down here, is as the urinary bladder fills, so as the bladder fills, it triggers these stretch receptors. And these stretch receptors are then going to send signals or action potentials. So coming in over here, so these are going to send action potentials to the spinal cord. So in other words, we have action potentials coming out of here when it gets full, and basically these signals come out to the spinal cord. And this is where the reflex comes in, is the spinal cord itself can actually send a signal back, and it'll send an action potential signal back. So this is going to be, of course, an action potential coming back to the internal urethral sphincter, telling it to relax. Now if it's telling it to relax, what it's actually using, so I'm going to go back down here under micturition, is it's using the parasympathetic network. So parasympathetic nervous system is what I'm going to put here. That is what's basically creating this action potential. Is this action potential is traveling through the parasympathetic network to get to the internal urethral sphincter, telling it to relax, right? Remember, parasympathetic is rest and digest, so this would be part of the rest. So this parasympathetic signal actually does two things. It relaxes the internal urethral sphincter, and at the same time it contracts the detrusor muscle. And these are actually a set of muscles, so I'm going to put muscles. The detrusor muscles are a set of muscles that overlie the bladder, and they actually run in several directions. So I'm just going to kind of overlay them here. Something like this. And so this red site here is detrusor muscles. Oops. And so this parasympathetic action potential that comes back and causes the internal urethral sphincter to relax also comes over here, and so the spinal cord is also telling the detrusor muscles to contract. So they're doing the opposite thing basically. So you're getting squeezing from the bladder and a relaxation of the sphincter at the bottom, allowing fluid to then begin to flow. Now, luckily this isn't the only thing that controls our urine flow, right? So the second part here, that's essentially the spinal cord reflex right there. But the second thing that controls the flow is our voluntary system, also called somatic. And our voluntary control is over the external urethral sphincter. And so that's the second muscle down here that I was talking about, right? That controls the flow out of the urethra. So in other words, your body will give you a signal, hey, we need to urinate. The stretch receptors in the bladder are showing that the bladder is full. Usually the stretch receptors in the bladder will trigger when you have anywhere from a cup to 2 cups of urine in your bladder. Typically around the 1 cup mark it'll start to trigger, and it'll relax these muscles down here, the internal urethral sphincter, and contract the detrusor muscles. That puts a little bit of pressure on your external urethral sphincter, and that's when you get your first real signal in your brain of, hey, you need to urinate. But you have control over that with your voluntary muscle of the external urethral sphincter. Now, there can be problems with the system, particularly in men. So in men, they of course have certain body parts that are in the way. So in this case, the prostate gland. So the prostate gland sits right about here, and you can see that it sits in between the internal and external sphincters. So this is prostate. Now, the problem for men is that as they get older, oftentimes the prostate begins to enlarge, and what it does is it will begin to push on the internal and the external sphincters, the muscles. And as it does, it can throw off how well they're contracting and whether or not they can actually contract completely around the urethra. And then this leads to urinary leakage, so they have problems with discharge of urine. Common problem among males as they get older, as their prostate gets larger. Okay, so at this point, I now want to show you a few slides that I have from my slide set, just to review some blood flow, and then we'll move on to filtration. You can see here from the slide that this is a slide showing the flow of blood coming into and out of the kidney. This is just for your review from the picture, because maybe the picture that I drew didn't make sense to you. One thing that I did not talk about on here is that the renal arteries and renal veins are sometimes ignored. There's one of them, I can't remember which, I think it's the renal arteries. Excuse me. So this is an image of the blood supply into the kidney. We drew this earlier. This is here for your review if you want to take a look at it. This is slide number 6 right now. One thing I do want to talk about, though, one topic here, is that if you recall, in this region we had segmental arteries and segmental veins. Sometimes I like to leave those in because it makes for a nice smooth transitional flow for arteries and veins. However, in some texts you'll see that I believe it's segmental arteries. They say segmental arteries do not exist. So one of the two does not exist if it's segmental arteries or segmental veins technically, but I like to just leave it in there because it makes it smoother for transition of inflow and outflow. It's all really the same. If you're asked that on an exam, just act like both the segmental arteries and segmental veins exist. It's all just naming nomenclature anyway. It doesn't matter that much. This picture here is kind of interesting. So this is actually a glomerulus with the Bowman's capsule removed, or glomerular capsule removed. So you can actually see this tight ball of capillaries, and we're going to get filtration coming across these small capillaries itself. You can also see some of the other tubules surrounding it here. So these are probably like distal and proximal convoluted tubules that are wrapping near the glomerulus here. This picture here shows a nephron shown in yellow here, and the tubules of the nephron shown in yellow, surrounded by the capillary beds that I spoke about earlier. So here you can see, for instance, the peritubular capillaries, the vasa recta down here. There's a transition, as you notice, of oxygenated blood over to deoxygenated as you move across the nephron. You can also see the inflow from the afferent arteriole over here and the outflow of the efferent arteriole through the glomerulus. I typically don't like drawings like this too much because it just gets so confusing with everything on here, but this is a good reference slide if my picture didn't do it for you, or if your drawing didn't work very well. I already spoke about micturition. That's just another word for urination. And so now we're going to talk about filtration. So remember, filtration is all about the glomerulus. So filtration takes place at the glomerulus, the capillary bed of the glomerulus. There are a couple other terms that I want you to be aware of here, so I'm going to transition back over to my whiteboard. Let me get my whiteboard up here. There we go. So one term that I want you to be aware of is fenestration. And I know we've talked about this briefly before. It can also be used in singular form, which is fenestre, but I almost always say fenestration. Fenestration refers to the pores in a capillary. And I'm going to say a capillary wall. Now, the reason why I'm bringing up fenestration again is because fenestration in the glomerulus is very high, very large. Okay, so glomerular fenestration. So glomerular fenestrae, so the individual holes in the glomerulus are 100 to 400 times more permeable to water and solutes than normal capillary beds. Okay, so again, the holes through the walls of the capillaries in the glomerulus, that make up the glomerulus, are 100 to 400 times more permeable to water and solutes than normal capillaries. So this is the point I'm trying to make, is this is an incredibly vacuous filtration. There's a lot of stuff that goes through this filtration, unlike other capillary beds in your bodies. Okay, still, the pore size, so a few facts about this. The pores, so the holes, right, they're still small enough to block certain things. They're able to block red blood cells, white blood cells, and most large proteins. So because of this, you should never see these in your urine, right? So these should not be in urine. And I'll give one caveat to that, and that is in very small numbers. So, in other words, you should not, in your urine, you should not see blood, you should not see white blood cells, and there should not be any detectable large proteins. The reason for this is because the glomerulus should not allow those through. If you see those in someone's urine or in your own, that is a sign of some sort of a problem, clearly. Okay. Anyway, this is what comes off of the glomerulus, you should understand, is, let me do this in blue. What comes off of the glomerulus is called filtrate, as you've heard me use many times before. This filtrate, I sometimes also called, another term for this I use is pre-urine. So you notice that I'm not using the term urine for what's coming off of the glomerulus, the filtrate coming off the glomerulus. That's because it's not truly urine yet. We don't get urine until the fluid actually passes out of the actual nephron itself. So once it leaves completely the nephron, then it's called urine. Until then, it's just filtrate or pre-urine. And the reason why is because the nephron continues to basically change what's in the filtrate. And so because it's changing so much, we can't call it urine. What exits the body or gets down to the bladder is urine by the time it gets there. Okay. So let's talk about another term here, and that is podocytes. You're going to see what are called podocytes. Podocytes are basically cells that line the glomerulus. And these cells are important because they can basically help to control the flow of filtrate. So basically they're one of the means of controlling how much urine is created. So control filtrate volume. And like I said, they are not the only means of controlling filtrate volume, but they are one means of controlling it. Okay, so with that, let's clear, and I want to take a look at the slide set again. So that's basically an overview of filtration. Now let's take a look at some of the cells involved in filtration. So you'll want to write these down definitely. These are important. So during filtration, so I just want to describe what the pictures are showing here. The upper left picture here, where my pointer is, shows a glomerulus. You can see blood flowing in through the afferent arteriole and then out through the efferent arteriole. Here, if you notice, the afferent and efferent, they have reversed from the way I normally draw it. It doesn't matter which way it's drawn. What you'd look for here are the arrows. So the arrows indicating blood flow indicate that blood flow is coming in up here, which makes this the afferent arteriole, and this one down here, blood flow coming out, makes this the efferent arteriole. In here we can see the glomerulus. You can also see the glomerular capsule surrounding the glomerulus. What will happen is as blood flows in, it's going to get filtered across the glomerulus. Now, if we zoom in on one, and then it's going to exit out the back here to the proximal convoluted tubule. If we zoom in on one of these tubes, which is what this second middle picture is a cross-sectional zoom in of part of the glomerulus. You can see here that we have a couple of layers here that you can see. First of all, there's an epithelial layer down here. And then on top of that, there are podocytes. And so these podocytes kind of look like almost like eggs that are stuck on top of or outside of the epithelial cells of the glomerulus. And then this third picture down here is an even closer view showing you a zoom in of the layers. So let's go ahead and talk about these layers that are actually making up the glomerulus. Okay, the first layer is this layer here. Basically looks like Swiss cheese. This is the endothelial layer. It's made up of endothelial cells. These cells have gaps between them, and these are called fenestrae. They make these little pores. That is our first filtration layer in the glomerulus. The second layer is shown here, and it's kind of this pink layer here. It's called the basal lamina. It's another name for it that you'll see right here. Let me get my pen out. Is the basement membrane. And so here they've turned it the glomerular basement membrane. That's fine. But this is the basal lamina or basement membrane. This also serves as a filter as well. We've talked about this layer before. It always comes underneath the endothelial layer. This layer has a tendency to block glycoproteins and negatively charged ions. So, or I should say negatively charged molecules. So let me make this very clear. It is blocking by size and by charge. That's how this blocking occurs. So in other words, anything that's too big is going to be blocked, and anything that has a large negative charge is going to be blocked also. And proteins, especially large proteins, have a negative charge and they are large. So they're not going to be able to get through this basement membrane if it's working correctly. Now, let me back up here just a moment. The endothelial layer with the fenestrae in it is also going to block. It primarily blocks by size, so it will block things like red blood cells, white blood cells, all the big things that we talked about. It will block some large proteins as well, but the basal lamina handles most of them. Then the third filtration layer, so there are three filtration layers really on this glomerulus. The third layer is shown here by these kind of finger projections. These are the pedicles or feet of the podocytes. So the cells up here that look like eggs, basically they have projections that come off of them that are called pedicles. Now for those of you that don't know, podo actually means foot, so these are foot cells. And they're named that way because they look like they have little feet extensions, or at least somebody thought they look like they have little feet extensions that come off of them. Now, these podocytes can be a little more, how do I say, they can be a little more selective in their filtration. They also filter by charge and by size, and so they don't allow proteins to go through. They block almost all proteins, and they block anything, not anything, but most things that have a negative charge. Okay, so again, I'm emphasizing here, all of these layers really block by size and charge. Endothelium is mostly by size, but the other two are by both size and charge. Not that it matters that much. They all basically block the same type of stuff. Now, In the specialization part of the podocytes is that there are slits between the feet of the podocytes. So if the tan portions here are the feet, there are slits between those feet. And the podocytes can actually open or close those slits. So that's what these filtration slits are that I'm mentioning over here. They're in between the foot pedicles, and the podocytes can control the width of those. So they can essentially control how much filtration goes through this last step. If we actually look at a cross-sectional picture of this, you can see it quite nicely. So let me describe what's happening here. Down at the bottom of this picture, you can see an erythrocyte, right? So let me get a highlighter here and I'll highlight these portions. So this down here is a red blood cell. Here is basically the lumen of the capillary in this red section here. Our first filtration layer is actually here. This section here is the endothelial layer. And you can actually see here one of the fenestrae, so a hole, a pore, between two of the endothelial cells. So this is what we're talking about as far as it being porous through this layer. Okay, from there we can see that fluid is going to be able to move through the filtration slits. Some will be able to move through here as well, just not as well, right? But we get a lot more flow through the fenestrae. Then the flow has to go through the basement membrane. And remember, there's blocking that's taking place here for negatively charged molecules and for proteins, right? So large proteins. And then up here, these are the podocyte feet. Okay, now you can see that the podocyte feet make filtration slits between them. So the filtration slit here is going to be this area in here. These are all filtration slits. And so as the filtrate comes off, it will be blocked by the feet. It has to squeeze through the slits. And then once it gets through the slits, it can then go off and eventually it will go to the proximal convoluted tubule eventually somewhere over here, right? But these are basically the three layers of control. So we have the endothelial layer here, the basement membrane here, and then the podocytes here that are controlling the width and size of these filtration slits. So each one of those is blocking by size and charge. Now here is one of the kind of confusing things sometimes for people. And that is that I've mentioned that they block things that are negatively charged. However, small negatively charged ions can get through. So for instance, chloride, so I'm just going to draw a chloride ion here. Chloride ions, negative charge, but chloride ions are very, very small. So they can get through the fenestrae, they can get through the basement membranes, and it can get through the slits. In other words, the thing I'm trying to let you know is that even though I've said this blocks by size and by charge, there are exceptions, especially to the charge portion of it. The size portion of it, typically nothing real big gets through, but small negatively charged molecules can get through, like chloride. Okay, so if you see exceptions like that, don't be surprised by it. Let's now move into what's described as glomerular filtration rate. So we usually abbreviate this as GFR, so glomerular filtration rate describes the flow rate of filtered fluid through the kidney. All right, so this is a description of now, you might ask yourself, why is this important? The flow rate of fluid through the kidney can be used to describe kidney function. So this describes kidney function. And with that, you can determine if someone has kidney disease. All right. So your GFR is actually a number, all right, and we'll talk about that number in just a moment. Now, the glomerular filtration rate is controlled by hydrostatic pressure, mostly, okay. And when I say hydrostatic pressure, what I'm actually referring to, that most people know this as, is blood pressure. So the higher your blood pressure, typically the greater your glomerular filtration rate. Now, this is very specific though. It's the blood pressure going into the nephron. So you could have high somatic blood pressure throughout your body, but have a lower blood pressure actually going into your nephron. There is some control over it actually. This glomerular filtration rate and hydrostatic pressure produce what we call the first filtrate. And typical volumes that we see on the amount of filtrate that's produced by the kidneys is approximately 125 milliliters per minute. And or this can be translated into 180 liters per day. And these are numbers for a young adult. So someone who's healthy. Now, I want you to look at these numbers and kind of be amazed by them, right? So think about this. You have about 4, maybe 5 liters of blood in your body. For a 180-pound man, that's about 5 liters of blood. And you're producing 180 liters per day of filtrate, of what we call pre-urine. That's a huge amount. That means your blood is getting filtered somewhere around 30 to 35 times per day, right? And that fluid is coming off as filtrate. Now, that means that you have to recapture that filtrate in order to not desiccate yourself within minutes, right, or within half an hour. So the thing that I'm saying is that 180 liters of filtrate coming off per day is a huge amount. We have to recapture that and reabsorb it. So the way that you're going to bring that back into your blood is through reabsorption. So there's a huge amount of reabsorption going on in the kidneys all the time. Now, let's take a look real quick at numbers for someone who might have kidney disease. So abnormal numbers here. So someone with kidney disease might produce maybe below 60 mils per minute. And this would be an indication of kidney disease. Typically, you're going to see numbers like this in someone who's older, so this number becomes lower. So as you get older, basically, your kidneys aren't as good at filtering, basically, is what that means. But we would look for numbers around 125 mils a minute. And they actually have ways of measuring this by putting things in your blood to basically take a look at your flow rate. So let's have a look here. Let me clear this off. And this picture here is basically a picture showing an example of how you can measure and see glomerular filtration rate. So basically what this is showing is, let me bring up my laser pointer. Up here on the top we have the afferent arteriole coming into the glomerulus, efferent out the bottom. you can see that in the blood we have these blue solutes and green proteins. That as the filtrate, or as the blood moves down into the glomerulus here, it gets filtered. What comes off are these smaller solutes. The proteins stay in the blood and continue away with the blood. This is going to produce this glomerular filtrate, and we call the amount that it makes the glomerular filtration rate. So let's take a look here at some of the things that control the glomerular filtration rate. First of all, the sympathetic nervous system can actually influence and control the glomerular filtration rate. And it does this by vasoconstricting the afferent arterioles. So remember, those are the arterioles leading into the nephron and into the glomerulus in particular. This vasoconstriction will lead to a decrease in the glomerular filtration rate because your pressure that you're putting through those or into the glomerulus is going to drop. Okay, this decreases your urine volume, but it increases your blood volume and blood pressure back in the somatic portion of your body. So in other words, the constriction of those vessels in turn backs up and increases the pressure and volume behind it, as well as the fact that you're not creating as much urine is also going to increase your blood pressure and blood volume. Now let's talk about this. So remember, the sympathetic nervous system is fight or flight. So this kind of makes sense that if you're in a fight or flight mode, you want more blood volume and more blood pressure. And so therefore, vasoconstricting these arterioles to restrict the movement of blood into the glomerulus kind of makes sense. Remember that all of this is auto-regulated, so you can't voluntarily regulate this. It's controlled by the sympathetic nervous system. And then there is also hormonal control over blood pressure. So the hormones that control blood pressure are shown down here. And so I'll talk about these just briefly. Basically, renin is produced by the kidneys. And you should probably write this down, that renin is in fact an enzyme, not a hormone. There's a distinction there. A lot of people call renin a hormone. It doesn't really matter too much, but it actually is an active enzyme. Renin participates in what's called the RAS system. So I'll write this down here. So RAS stands for renin, angiotensin, aldosterone system. I often call it the RAS system, and I know I'm duplicating system, but whatever. So R is renin. Renin is released by the kidneys. So renin is released when blood pressure is low, and it's released by the kidneys. And renin is, like I mentioned, an enzyme. That enzyme will then cleave angiotensinogen into angiotensin, and I'll go through this later. But then angiotensin then causes the release of aldosterone from the adrenal glands. Now, aldosterone in turn is going to increase blood pressure. So when blood pressure is low, then essentially the RAS system kicks in order to increase blood pressure. And it does this by pulling sodium back into the blood from the filtrate. And again, I'll talk about this more later on. So let's take a look here at how some of this works, or how some of these systems work as far as regulating the GFR. So if we have a decrease in blood pressure, this is going to be detected by baroreceptors, and they in turn are then going to trigger an increase in sympathetic nervous activity. So drop in blood pressure leads to then sympathetic nervous activity to increase. That causes an increase in heart rate and cardiac output. But we can also get vasoconstriction. The vasoconstriction within the kidneys, like we were just talking about, leads to an increase in total peripheral resistance, which increases blood pressure across the entire body. This then, because we're vasoconstricting the blood going into the afferent arterioles, this decreases the glomerular filtration rate, because basically what we're doing is we're cutting off blood flow or limiting blood flow to the glomerulus. So that's going to decrease our glomerular filtration rate, which decreases urine output, and this in turn increases blood volume. So we get both, when we have low blood pressure, we get an increase in both resistance and blood volume in order to push pressure back up. Okay, so let's talk about one of the major terms. So we talked about filtration and how that happens across the nephron, right, or across the glomerulus. Now let's take a look at reabsorption. So reabsorption, if you recall, is the return of solutes in water back into the blood. And again, this is from the filtrate. Remember that we produce approximately 180 liters of filtrate per day. However, we only produce about approximately 1 to 2 liters of urine per day. So what that means is that we need to reabsorb approximately, if you subtract these two numbers, 178 to 179 liters of filtrate per day. In other words, your filtration system is incredibly inefficient in many ways. You're producing 180 liters and then you're having to reabsorb 178 to 179 liters back into your blood. So that's a huge amount, right? So there's a huge amount of reabsorption that takes place every day within your nephrons. Now, as one other distinction that I want to make here is that your obligatory urine production is approximately 400 milliliters per day. In other words, what that means, obligatory means that you have to produce it. So every single day, just to handle your nitrogenous waste, you need to make at least 400 mils per day. Now we produce about 1 to 2 liters because we drink quite a bit of water, so it helps to dilute that out. But if you're stuck in the desert, this is one of the problems, and you don't have any water, you're still going to be peeing about 400 mils of fluid out per day that you're going to have to replace sometime in some way. So let's talk about where reabsorption takes place within the nephron. It actually takes place throughout all the tubules of the nephron. But here I'm going to focus for today on the proximal convoluted tubule reabsorption. So let's take a look at how proximal convoluted tubule reabsorption actually takes place. And to do this, I'm going to draw an illustration here. So imagine here I have a cell. Get this going here. I have a cell of the lining of the proximal convoluted tubule. Okay, so this is a PCT cell, and I'll put another cell over here just to complete this picture, right? So here, if you can imagine, up on top we have this is the lumen of the proximal convoluted tubule. So within the lumen, we have flowing along the top here is filtrate. So this is filtrate within the tubule, within the lumen of the tubule. This filtrate, just so you're aware, and you can write this down, it has too much of everything in it. It has too much water, it has too much glucose, it has too much salt, and so we need to get that back, okay? And we want to reabsorb that into the body. So on the other side of the proximal convoluted cells, at some point we have blood supply. And so we have blood flowing along over here, where we'll recapture everything, okay? Now what we need to do is get the stuff from the filtrate, we need to get it to move across the cells and into the blood. All right, so that's what we're going to talk about, how that actually takes place. First of all, let's talk about sodium. So we're going to look at the different molecules and how they move. There's too much sodium in the filtrate. Sodium is going to move through, let me draw this in black here. Basically just some sodium proteins, where basically sodium can just flow through these proteins by diffusion because we have a high amounts of sodium in the cell, or excuse me, in the lumen where the filtrate is. So this sodium is just going to naturally diffuse, so this is going to travel by diffusion into the cell. Now, this of course leads to more sodium in the cell, but we can actually use this high to low concentration to also drive some other things that we want in the cell as well. So over here, we have what's called a sodium glucose pump. So here we also have too much glucose. Remember, we want to recapture all of that out in the filtrate. Remember that everything that I'm drawing in blue here is in this filtrate. Now glucose is a little bit different because it doesn't want to go into the cells because the cells have a pretty good amount of glucose in them. So instead what it does is it's going to hitch a ride through a co-transporter, if you recall. Remember, this is going to be a symporter, where as sodium moves down its concentration gradient, it pulls glucose with it up its concentration gradient. So now we have more glucose in the cell, as well as some more sodium moving into the cell. Now you can see that concentration levels in the cell begin to rise of both of these, and so we need a way to get rid of these. So luckily at the basilar side of the cell, there is basically a glucose transporter that allows glucose to move down its concentration gradient. So now we're getting glucose back into the blood, which is where we want it. This type of movement system will eliminate almost all glucose from the filtrate eventually. Now we also need to move the sodium out so that we still have a sodium concentration gradient so that we can drive all of this. And the way that your body does this is it uses the sodium-potassium pumps on the bottom of the cell here. So these down here are sodium-potassium pumps. If you remember those sodium-potassium pumps, it's going to move 3 for 2, right? I'm not going to draw that in here, but it's basically going to exchange sodium for potassium, and so now we're winding up with sodium in our bloodstream rather than in the filtrate. Now, the benefit of this is that there's a lot of solutes and water that like to chase after sodium. So you can see that through diffusion and through co-transportation, we're getting sodium and glucose where we want them. They are now in the blood. But there are other things that will chase after the sodium. So for instance, we have too much chloride also in the filtrate up here. Luckily, chloride likes to chase after the positive charge of the sodium. So chloride will actually move on its own. Usually it has a transport here as well, but that doesn't matter as much. So this is going to bring chloride into the cell for free because we moved sodium, and then chloride will eventually chase sodium out into the blood as well. So we get the movement of chloride for free. The other thing that likes to chase sodium as well is we will get some movement of water as well. And so this water is going to chase after the sodium, and eventually it's also going to wind up in the blood also. So you can see that simply by moving sodium, we're actually getting the movement of 4 molecules that we want to move if we include sodium. And basically, as long as we move sodium into the blood, we're getting chloride, glucose, and water all chasing it. In the proximal convoluted tubule, we will reabsorb or recapture about 2/3 of our sodium, our chloride, and our water. Okay, so we're getting a reabsorption of 2/3 of all of those. Now, so in other words, there's a huge amount of reabsorption taking place in the proximal convoluted tubule. Another thing I would add up here too is that this reabsorption is non-stop. It's ongoing, just constantly, never stops. Also, there's no hormone control over this. It just goes all the time. Now, when we talk about reabsorption numbers here, so let's assign some numbers to this real quick. So if we're absorbing 2/3 of our water in the proximal convoluted tubule, that's going to approximately equal about 120 liters per day. So you can see that we've already taken care of about 120 liters of the 180 liters that we need to reabsorb, right, or 179 or so. We also reabsorb approximately 90% of our potassium, which I did not show in this picture here, is reabsorbed here. Now, the reason I didn't show potassium is simply because there's not a lot of stuff that chases potassium, but you should understand, like if we were to add it here, I'm going to do it in a different color to show that it's not chasing sodium. But basically, if we had potassium out here, it's also going to move into these cells and then eventually into the blood also at a rate of about 90%. Now, the last thing I want to add here is that you actually have to expend a lot of energy in order to maintain this reabsorption rate. We expend about 6% of our basal metabolic rate just in this area. So for instance, running the sodium potassium pumps. In other words, so to be clear about what I'm saying here, out of every 100 calories you eat, 6 of those calories go towards just driving this reabsorption in order to bring your filtrate level down. It's a huge amount of calories. Most of those calories are being used to run these sodium potassium pumps down here. But I'm just trying to make the point that this is a lot of energy that you're spending on this. Okay, so finally here, I want to just run through a couple of slides that I have at the very end. They basically just reiterate what we just went over, but I'm going to show them to you. They're here for your reference. This is slide 16 on the current slide set. You can see that here, this is showing the Bowman's capsule with glomerulus and the filtration that's taking place across the Bowman's capsule, and then the reabsorption that's happening out here in the proximal convoluted tubule, which is what we were just talking about. Here, this slide is describing what I just basically went through on the cell. So it talks about the sodium potassium pumps on the basilar side of the proximal convoluted tubule cells. It talks about the gradient that's created, the fact that chloride and water are going to follow and chase after sodium, and that we have a cotransporter in order to get glucose back from the filtrate back into the blood. Of course, I already went through all of this, so you can just read this. Then this final slide just goes over essentially what I was just talking about. 2/3 of our sodium chloride and water are brought back in from the filtrate in the proximal convoluted tubule. As far as water goes, this is about 120 liters of water per day. About 90% of our potassium reabsorption also takes place in the proximal convoluted tubule. Again, this reabsorption is non-stop. There's no hormonal control over it. And we spend about 6% of our overall calories just in driving this reabsorption. So it's a huge amount again. And then this is the last slide here. Okay, so this slide just shows the reabsorption of water and salts basically. And that's it. Thank you very much.